Great. Well, good afternoon, everyone. Thank you for joining us once again for our weekly Wednesday webinar series. Today's webinar is on, I have a different title than on your presentation, but fishing for answers to hemostatic and thrombotic disease, gene editing in zebrafish. But we have a more thought out title right here on Dr. Shavit's presentation.
Yep. I just forgot to change it.
Oh, that's okay. I should have asked earlier. My name is also Mirtha, and I am the moderator for this series. At any point during the webinar, if you'd like to ask a question, please use the Q&A feature at the bottom of your screen. And if you're joining us from one of our live streaming platforms, you can always ask your questions in the chat or in the comments. We have staff monitoring these questions, which we will pose to our speaker after the presentation. This webinar is being recorded and will be available to the community beginning on Friday, February 24th. This is our last webinar for the month of February, which is really exciting. I'm joined today by Dr. Jordan Shavit, Professor of Pediatrics and Human Genetics at the University of Michigan. Thank you for taking the time to join us today. I'll go ahead and turn it over to you to get us started.
All right, thank you. It's a great pleasure to be here. The title is a little bit different, but it's basically saying the same thing. As I said, I forgot to change this one. So I'm going to talk to you today about genome editing in zebrafish. This is just a slide I'd like to start with, which you can see live zebrafish swimming. I find it very soothing. And then you can see my collection of Go Blue zebrafish. So as I said, I'm going to talk to you first about genome editing to introduce it, the mechanisms behind it, the applications, how it's used in animal models, and then I'll talk about how we use it in zebrafish to produce models of bleeding disorders, to analyze human disease mutations and study large populations. I'm not going to spend a lot of time on this slide. I think this is familiar to the community. On the left, there's, my lab studies both thrombosis and bleeding disorders. Thrombosis is pathologic blood clotting, of course, and it can happen as a DVT, deep vein thrombosis, or stroke and heart attack are both forms of thrombosis in the arterial system. What we're going to focus today is on bleeding disorders. We're very familiar with hemophilia and von Willebrand disease, and my interest is in the genetics of these disorders. And then also we have an interest in how genetics influences hemorrhage that may be in patients who don't have a known bleeding disorder. So of course, I think this audience is familiar that hemophilia is an inherited bleeding disorder, usually passed through the mother. It's sex-linked, meaning it's on the X chromosome. And then there's von Willebrand disease, which is autosomal. It can be it's more regularly seen in both males and females and passed by the mother or the father. So I want to, not assume that people, I don't know exactly how much people understand about genetics and genes. So I'm going to go to the basics here. That DNA is our genetic code, right? Probably most people know that. It's 3 billion letters long, and it's packaged into chromosomes, which you can see in this picture here. And these chromosomes come in pairs. We have 23 pairs of these chromosomes, and you can see them here starting at chromosome 1, 2, 3, etc. This is the X chromosome here, and this is the Y chromosome here. And these are packaged very tightly. They're in the nucleus. They're all wrapped up so they can fit in the nucleus. But if you pull it out into a single string of DNA, I forget the length, but it's very long, something like yards or miles long. And it's made up of these, what we call nucleotides or base pairs. And that's the genetic code. And they come in A's, C's, T's, and G's. So just 4 letters to that code. And you can see that DNA is what's called a double helix. It looks like a ladder that's wound up. This is what it looks like unwound. And again, you can see the code A, G, C, T, etc. And so these codes make up genes. And actually, the genes part of DNA is relatively small. It's probably 1/1000th of or so. It's about actually no 1/100th of that 3 billion letters. And so the majority is other DNA that we don't fully understand what it does. But let's talk about genetic diseases. So a genetic disease like hemophilia or von Willebrand disease is characterized by a primary disease mutation in DNA. That mutation is in genes we know about, like factor VIII and factor IX for hemophilia, and then von Willebrand factor for von Willebrand disease. Now, that's not the whole story. You may know people who are in the same family with the same disease. This is especially true in von Willebrand disease. And they may have very different symptoms where somebody bleeds a lot and somebody doesn't bleed much at all. And this is due to a number of things. One is environment. So injuries, surgery, how one behaves, how the environment impacts you can affect that. But also genetics are important. We have 20,000 genes, and we all have variation across those genes that's inherited separately from this primary disease mutation. These are called modifier genes, and they can impact how much bleeding one has. And I think it's illustrated nicely in this pedigree. This is actually from a thrombosis disorder, factor V Leiden, but I think it's illustrative. So factor V Leiden is a mutation in factor V that puts individuals at risk for thrombosis. And what you can see here is that there are 15 individuals who are shaded on the right, meaning that they have this factor V Leiden gene mutation. But of those, only 5 are also shaded on the left, meaning that they've had an episode of thrombosis. You can see them here, 1, 2, 3, 4, 5. So those 5, so what's happening here is that there are modifier genes being inherited separately from factor V that may be protecting some individuals or putting others more at risk. And we're interested in getting at those modifiers. If we understand them, we could have a better basis for predicting who's at more risk of bleeding or thrombosis. Now, genetic risk factors for acquired disease are also in my interest, as I mentioned earlier, seeing why some people who have trauma or surgery might bleed more than others, even if they don't have a clear inherited bleeding disorder. So to study this, the power of genetic studies is to have very large pedigrees. Human pedigrees can be quite large, but usually they're not terribly big in terms of extended pedigrees that we can map diseases. And that's why we turn to animal models. With mice, we can make very large pedigrees. Actually, they're technically infinite if you're using certain strains of mice, but they can get very expensive to work with. And that's why we use zebrafish. A single mating pair of zebrafish can produce 200 or 300 offspring, and they can do that on a weekly basis. Now, this is a figure I have for how we've been able to study the genetics of zebrafish, but I've added in some time points related to mice as well. You can see here that before we could actually go in and target a specific gene, what we want to do is we want to be able to make mutations in genes so we can make a model of that disease in animals and then study that to do things we can't do in humans. Now, to be able to do that in a targeted way, to pick specific genes, hasn't been around till recently. The first mouse mutants were identified just spontaneously back in the early 20th century. These were things that were very obvious to see, like coat color or size or tail length. In the early 1980s, people started to do that with zebrafish as well, to see different sizes of fish or fish that had no pigment, et cetera. And then in the early 1990s, we first got the ability to do targeted gene mutations in mice. And these are called knockouts, in which you take away the gene, and now that mouse has a human disorder. And mice have been made for hemophilia A and hemophilia B. We were able to do something called chemical mutagenesis in fish that induces random mutations, and we could do that and then look for symptoms or what we call phenotypes in our areas of interest. We could look for small fish or big fish. Nobody was doing it at the time, but we could look for example, fish that bleed a lot. We could look for what some people did is they look for fish that didn't have blood cells. But that wasn't a targeted way of doing it. We couldn't really do targeted gene mutations easily in fish until genome editing came along. And that started about 15 years ago with something called zinc finger nucleases. And I'll talk about those a little bit because I think they're important. There actually is a clinical trial going on with zinc finger nucleases for hemophilia. Zinc finger nucleases are very hard to make and they don't work very well. They're so 2008. We don't use those anymore. Talons came along after, and they worked really well, but were still kind of hard to make. And then Crispr, which many of you may have heard of, came along about, 10 years ago, and revolutionized the field. So I'll come back to explain those in a little bit, but let's talk about modeling genetic disorders and what I mentioned to you, gene knockouts. These first came in mice, and they were pretty difficult to make. This is in the late 80s, early 90s. And what they did is, what one does is takes embryonic stem cells, which come from an embryo, and culture them. And in culture, you can then target the gene of interest to knock it out. You then take those cells and inject them into embryos. Often different colored mice are used, black mice here, white mice here, injected into embryos. You get these chimeric mice. You can see they're a mixture of black and white, and then they're mated to get black mice and white mice. And then these have the gene of interest knocked out. So for example, here you can see a sibling with its knockout pair, and you see the knockout weighs more than the sibling. Now with CRISPR or other genome editing factors, we can do this in a single step, and I'll show you how we do that later, where you simply inject it into mouse embryos or fish embryos and get a knockout. So how do we intend to use gene therapy for inherited disorders? Well, we all know, or I think most of the people on this call know, that there is now gene therapy for hemophilia. It's been approved, hemophilia B in the US, hemophilia A and B in Europe, and we're going to see this more and more. This hemophilia is done by packaging the corrected gene into a virus, adeno-associated virus, and that is infused into a patient. It then goes to cells, particularly in the liver, injects its DNA into the nuclease, and has that copy sitting there. But it's not targeted. It's just putting this extra copy of DNA in there, which sits around, which can either sit outside of the genome or can insert itself into the genome and cause problems. That is always a concern. Gene editing would be very precise targeting of that DNA into a specific site in the genome to fix a defect. And it is, will also be used with adeno-associated virus, who just simply package this into the virus. And that can help give a little bit better targeting. So this is what genome editing nuclease looks like. It's usually a protein that will then has a nuclease protein. So this protein binds to DNA. This is a nuclease that's like a pair of scissors that will cut the DNA. So this is supposed to animate in later. Ignore this for now. We'll come back to that. What happens is, so for zinc fingers, these are made of modules of individual proteins that can bind to 3 base pairs or nucleotides in the genome. And they are linked up with other modules that can bind to different base pairs. So you've got something here that targets 9 base pairs. This one targets 9 base pairs, and it will only cut when it has this nuclease protein that functions as a dimer, as 2 proteins, and it will only cut in that case. Talons, same thing, but each module binds to 1 base pair, and here's a string of 12, and here's a string of 12. So this is a recognition sequence of 18. This is 24 total, and that's good enough. Even in a genome of 3 billion base pairs, that's enough to find a unique site. CRISPR works similarly in concept, but it uses RNA. So a piece of RNA that's 18 to 20 base pairs long will find its target site, and then it binds to a nuclease protein, Cas9, that will do the cutting. And what happens is, with any of these, they cut the DNA to make what we call a double strand break, cuts both of both sides of the ladder, and that can lead to either a deletion or an insertion of DNA, which will typically mess up a gene. Or you can put in a little piece of DNA that will do a correction. And you can see this, if you can see that little red nucleotide there, that's a correction to a mutation. So that's one way you might think of fixing an inherited disorder like hemophilia. Or you can put in a larger donor that has a whole gene. Here it's with green fluorescent protein, which you can see has been done in mice. And you can see this green fluorescence. Of course, we don't want to do this with green fluorescence. We want to do this with, say, factor VIII, factor IX, or possibly von Willebrand factor. So I think it's helpful to see this multiple times because this may be new to a lot of you. This is a video from the Mayo Clinic. It's about 1.5 minutes, and I'm going to go ahead and play it. Actually, I don't know if it's going to come through to you. Let me do something. I'm going to stop sharing for just a moment. And I'm going to share again, but I'm going to have share my sound here. Hopefully the sound will come through better now. So it's playing. Alston, tell me.
In a document, if we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, that function is taken on by a system called CRISPR-Cas9. CRISPR is short for clustered, regularly interspaced short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA-cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut-and-paste tool for DNA editing. In the future, scientists hope to use CRISPR-Cas9 to develop critical advances in patient care or even cure lifelong inherited diseases.
Okay. All right, can you still hear me?
Yes.
Okay, great. So I thought that was a nice video to hammer the point home. I also want to give you a little bit more background on where CRISPR came from. It's not like somebody, thought, hey, I can take this little piece of RNA and I can take this protein, I can put them together and I can get genome editing. It came from nature. And actually all the other genome editing tools came from nature. So I'd like to point out that science is a team sport. I compare it to, Willy Wonka or Charlie and the Chocolate Factory. If you remember, Veruca Salt wanted to win a golden ticket, so her father had the money to do it, and he got all of his workers to work as a team. Not really a kind of exploitation of a team, but they all work together, and you see all these individuals randomly opening up bars of chocolate to look for that golden ticket, and one finds the golden ticket. And so science is sort of like that. We are all working on all sorts of different interesting scientific problems, but sometimes they may sound a little odd to you. Like, why would somebody do that? Well, sometimes that can lead to something that's clinical therapeutic. And so what people were working on for CRISPR is they were working on bacterial immunity. And you might, some of you might think, hey, I didn't even know bacteria had an immune system. You might think that's strange. Why do we need to work on that? There's all sorts of reasons, understanding how bacteria work, since bacteria are pathogens, is important. But nobody ever dreamed that working on bacterial immunity would lead to a fantastic tool for genome editing. But it did. So bacteriophage are like viruses to bacteria. They land on bacteria, they inject their DNA, and bacteria have evolved ways to protect themselves from that. That's what CRISPR is. It's a series of DNA that DNA sequences that over evolutionary time, bacteria has matched to these bacteriophage DNA. So when bacteriophage inject their DNA, they can match it with CRISPR, and then they can go and chop that DNA up and protect themselves from that DNA. So we've taken this and repurposed it into a medical tool. And so I think it's important that we have many, many labs working on a lot of different things that don't seem obviously related to human health, but can lead to that way. So as I mentioned, gene therapy for inherited disorders is currently with AAV that injects DNA randomly into cells, but we can put these together to inject gene editing materials to then target a particular gene. The first time I saw this done was in mice by Kathy High's group back in 2010. Remember, the idea is that you cut the gene, you put in a repair template, and that leads to a fix for the gene. And their approach was clever in that they noted that hemophilia B has a particular clustering of mutations. So for those of you not familiar with genes, genes are divided into exons and introns. Factor IX has 8 exons, and that's where all the business part is. That's where all the coding for the protein is. And they noted that 95% of factor IX mutations are in exons 2 through 8. And exon 1 is usually normal. So what they realized is that they could take a gene therapy approach where they take their repair template is normal exons 2 through 8, and they could target it with a single zinc finger nuclease to this one part between exon 1 and 2, known as the intron. And now they could get that inserted, and what you get is a newly reconstituted factor 9 gene. This becomes extraneous, but this here will produce factor 9, and they showed in the mice that it could produce factor 9 and lead to much better clotting outcomes for hemophilia B mice. And the reason why this is important is because when you're designing these tools, if in the future you're designing this for a patient, at least back in the days of zinc finger nucleases, it was, I told you, really hard to make these. And so you can't imagine being able to make everybody's own individual zinc finger nuclease to target their mutation in exon 2 or 5 or 8. You would really need something like this. And then this could be a gene therapy approach that would work for 95% of individuals with factor 9. Now, gene therapy has different routes in other ways. One is to package the reagents into a vector and inject it into a patient, have it go to the liver, which is what AAV does right now. But there are other methods, and that would be to do it in cells. This is called in vivo, to do it in cells ex vivo. So ex vivo, you take out cells, typically bone marrow cells would be something that's done, and modify the cells and then put them back into the patient. And that has advantages in a number of ways. It's easier to target a specific cell type. It's easier to target a specific location of the genome. If you know how to culture those cells and grow them up, you can make a lot more of them before putting them back. This takes a long time, right? So, if you go back, the first AAV gene therapy trial was before 2000, and it's ramped up. And we know over the last 10 years, we've had a number of AAV trials for hemophilia A and hemophilia B. And this shows the same thing. So it's going to take some time, and it'll be interesting to see whether, whether gene therapy can, genome editing gene therapy, how quickly that will go. So how does this work? How do we use it in animals? I'm showing you for a zebrafish. This is how it's done for mice. What we do is we take the genome editing reagents that might be zinc finger nucleases, as shown here. We did this for antithrombin about 10 years ago. Now we solely use CRISPR. We take these, we inject them into a single cell embryo, and we grow that up, and we screen for a knockout. For antithrombin, because zinc finger nucleases didn't work well, we had to inject 2,000 embryos. We got 46 fish that we made in, and we got 4 lines with different mutations. We got a 4 base pair insertion, a 6 base pair deletion, a 22 base pair deletion, and a 90 base pair deletion. And when you get these deletions, the genetic code is a multiple of 3. So every 3 base pairs codes for a amino acid in a protein. So if you get something that is not a multiple of 3, that shifts the code and effectively is a knockout. So now I'm going to switch gears. I've talked to you about genome editing. I'm going to tell you how we apply this in zebrafish. So first I want to introduce coagulation in zebrafish because you might wonder why we're using a fish. Well, a fish is a vertebrate like mice and humans are, and coagulation is very similar to mammals. If we look at the genome, the whole genome of fish has been sequenced, and they have the same genes, and the functions are similar. Advantages of the zebrafish model include that the ability to produce hundreds of offspring, which I told you about already. And then they go from a single cell embryo to a free-swimming larva in 3 days. And this free-swimming larva has nearly every major organ system, including a beating heart and circulating blood and an intact coagulation system. They're also transparent, so we can see that coagulation very easily. We can keep them in Petri dishes. We can keep about 100 in each Petri dish, and they don't need food for the first week of life, so it's very easy to work with them. And then they become adults at about 2 months of age. So we can put them into Multiwell plates. This is like a plate full of mini test tubes. You can see them in here, and we can test new drugs on them to find drugs that will treat the models we make. So we've made a number of bleeding and clotting models, and we're using this kind of what called chemical screens to find new drugs to treat them. One thing we do to test how clotting is working in our mutants is we hit them with a laser. We injure the vessel, and that induces the formation of a blood clot. And you can see this here. And this is what it looks like when clotting is working. You watch that little blip there was the laser hitting, and you'll see a blood clot that's growing that will eventually obstruct circulation. You can tag cells and molecules in zebrafish that are circulating with fluorescent proteins. Remember those fluorescent mice I showed you before? We can do the same thing here. So we've tagged fibrinogen with GFP, green fluorescent protein, and you can see it makes a green fluorescent clot. And in the that's in the venous system. In the arterial system, what we see is another group has tagged the equivalent of platelets. They're called thrombocytes, and they tag those with GFP. And you can see when we hit with the laser, we get these green fluorescent platelet clots. And this is like what you see in stroke and heart attack, platelet clumps in the brain or in the heart. And in deep vein thrombosis, it's typically fibrin-rich clots. So we're seeing the same kind of symptoms in a fish that we see in humans. So we've also done some imaging of adult blood clots. This was work done by a former postdoctoral fellow in the lab, Megan Rost Griffin. And what she did was she worked with John Weisel at Penn. We took adult whole blood from fish and tested it. We let it clot, and then John's group did scanning electron microscopy. And what's interesting is he had done this previously with mice and with humans. And what you can see is the red blood cells pack into these polyhedrocytes. It's very tight packing. You can see these shapes here. And what was surprising to us is that what I didn't mention to you earlier is that zebrafish cells are different than mammals. They have nuclei. And we thought the packing would be different, but you can't tell the difference between these. This is a zebrafish, this is a mouse, and this is a human, and they all look alike. So some of the things we do to find new activators of clotting using those multi-well plates I showed you earlier is we have our fish with our circulating fluorescent cells, the thrombocytes and red blood cells, and we look for fish here. If you look down here with clots or without blood clots, you can see this fish has a big clot, and we found a compound that was promoting this clotting that potentially we hope might be useful as a treatment for bleeding disorders. And you can see what that looks like in by scanning electron microscopy. You can see that when we treat with this compound, we get these polyhedrocytes again, and what we think is fibrin formation here. So what we've done is, this is the coagulation cascade. Here's factor 8 and factor 9 and other coagulation factors you might know, like factor 10 and prothrombin, fibrinogen, platelets, von Willebrand factor. We've used genome editing now to knock out all of these coagulation factors and study them. I'm going to just talk to you today about knockout of factor 10 as representative of something that when you knock it out gives the fish a bleeding disorder. So we knocked out all these factors, and with the phenotype I'm going to show you now, the effects are similar in the extrinsic pathway, which is tissue factor and factor 7, as well as the common pathway, factor 10, factor 5, and thrombin. And what happens is when we make knockouts for these, we represent it as plus minus. Minus, one plus means one copy of the gene is intact. Minus means that the other copy of the gene is not intact. And when we mate them, what we get is we get some fish that are wild type, we call it 2 normal copies, some that are carriers, and some that are complete losses, and they have factor 10 deficiency. And this is what their survival looks like. At about 2, they survive, actually surprisingly long, until 2 to 3 months of age into adulthood, and then they start dying. And what do they die of? They die of hemorrhage. Not surprising. At 1 month of age, we can start to see hemorrhage. You see this red here? This is blood pooling in the head, in the brain specifically. You can see it here. We've done histology on them, and you can see these arrows are pointing to red blood cell accumulation. This is pools of red blood cells in the brain. And then we see muscle bleeding too, pools of red blood cells in the muscle, not unlike what we see in hemophilia. So I already showed you this, that we can make clots using laser. And we did this on our factor 10 mutant fish. And what we found is that when we injure them, the factor 10 plus fish clot normally. They form a clot within about 60 seconds. The carriers do the same. But the homozygous mutants, the ones that have complete factor 10 deficiency, they don't clot at all. we stopped this at 2 minutes because if they don't clot at that time, they're not going to clot at all. So this is showing that they have a bleeding effect. And what was surprising, the reason this was surprising to us, actually, I'll come back to that in a moment. But one thing we can do with these is we partnered with Flora Pavandi in Italy, and we looked, this is the factor 10 gene. This is the domains known for the gene. This is the amino acid sequence. We're comparing fish to humans, to mouse, to chickens. And what you can see here is we've picked certain amino acids that are mutated in factor 10 deficiency that Dr. Pavandi had found mutations that hadn't been studied well. And we found ones that they have the same amino acid. If you look down here, they have the same amino acid in all these species. Well, not all of them, but most of them. And so we took those ones that were conserved from fish to mouse to humans, and we tested them. And you can see here, we engineered those mutations into zebrafish factor 10. And you can see here, remember, they don't clot with laser injury when they don't have factor 10, but we're able to put back in a normal copy of factor 10, and then they clot just fine. And here we can see how well they clot when they have these various mutations. So it's a way of, another way of looking at human mutations in an in vivo situation rather than just in a test tube in the laboratory. And so we found some interesting differences between the common pathway in mouse versus fish. So in fish, if you take all these common pathway mutants, factor 10, factor 5, and factor 2, or thrombin, they all have the same phenotype. They have bleeding primarily in the brain and also in the muscle. And they live into adulthood and don't die until they're 6 months of age or so. In mice, it's very different. Mice actually, they have bleeding as embryos, and most of them die as embryos. And if they survive to birth, they die shortly after birth. So there's something about fish that when they lose these coagulation factors, they have the same overall effect, the bleeding effect, but they survive longer, which is interesting. And if we study that, we hope that we can, find out why that is, and study that further and get some ideas about that there may be some species-specific differences between humans and fish. And if we can exploit those, we might be able to treat bleeding disorders with that approach. All right, so I'm going to switch gears here a little bit, and I'm going to talk about how we've partnered with people who look at large human populations and use their zebrafish to find genes that are involved in clotting disorders. So this is actually thrombosis that we're looking at now, but we can apply this to bleeding disorders as well. So this is what's known as the Global Biobank Meta-Analysis Initiative. And what you can see is it's a number of biobanks. Biobanks are when you have a place like we have one here at the University of Michigan, for example, where we collect electronic medical record with consent, of course, electronic medical records on patients, and then we also get their DNA and sequence their genome. And for those who have consented to this, we put them into our biobank. You can see here the Michigan Genomics Initiative has 55,000 individuals. And there's some that are smaller, that are 7,000, and there's some that are huge, like the UK Biobank has 500,000 individuals. It's a biobank that's across the United Kingdom. And all these biobanks have partnered together for this, what's called a meta-analysis, to look at all their data together. And so we partnered with a group at Michigan who works with them. And they partnered with a subset of biobanks to look at venous thromboembolism or venous thrombosis. And they had 28,000 from this group. They had 1 million controls, so people who did not have thrombosis, and 28,000 who did have thrombosis. And then what they did is, this is getting a little complicated even for me. They do a number of algorithms to look through the data. And all these different algorithms have different ways of looking across the whole genome and saying, okay, and so what this is you're taking these two populations and you're saying, okay, what is genetically different about this group versus this group? And if we can find those differences, we find certain genes that have variation always in this group or always in this group, we can say that gene is somehow related to thrombosis or prevention of thrombosis. So they use these algorithms to look through, and they came up with a list of 35 genes that are suspected to be related to thrombosis, that there was variation in these genes that was enriched in the thrombosis population. Now, the problem is when you have that, it's just a guess as to whether these genes are truly associated with thrombosis. You have to do another test. You need some kind of laboratory test to say that, yes, this gene is associated with coagulation. Now, one thing that was really interesting, and what these gray bars mean, is it means that, for example, for this gene here, it was associated with this algorithm. It was positive in this algorithm, this one, this one, this one, this one. So that's 5. This gene only had a positive in one of the algorithms, right? So 5 versus 4 versus 3 or 2, 1 lines of evidence. So the genes that are more likely to be involved in thrombosis are the ones towards this side, and the ones that are less likely are this side. Now, when they came to me and they said, well, can you model this in your zebrafish? And we could, we could use CRISPR to model these, but 35 genes is still a lot to do. I've showed you, we've made about 10 knockouts in the coagulation cascade over 10 years. So to do 35 rapidly was pretty tough, and it's possible that we could find that none of them are associated. They're all false, what we call false positives. But I was encouraged when I looked at the names of the genes, and if some of you may have been looking and recognizing some of these genes. These genes here, shaded in yellow, are all known coagulation factors or known genes involved in clotting in some way. Look at them here: factor 5, protein S, factor 11, prothrombin, fibrinogen genes, von Willebrand factor, factor 7, factor 10. You get the idea. And some other genes, protein C receptor, and other genes you may not have heard of, but are known to be associated with thrombosis. And they're all shifted to the left, right? So most of them are at least 3 lines of evidence. So that said to me, okay, this system is pretty good, because we already know these are involved in clotting or thrombosis. So those are already real hits. But what about these other genes in between? So we picked a number of them. We actually decided to pick some genes even down here to study them. These genes here that are dual colored are ones that were what we call positive controls that we already knew were involved. And then we picked some that we had no idea whether they were involved in clotting. Oh, and one more, actually, this one that I'm going to show you the data for. So what we do is we simply order these guide RNAs. We can now just do this using a computer. This is led by a medical student, Queen Zhao, and working with my lab manager, Katie Richter, and a postdoctoral fellow, Xinge Yu. And what they did is they simply look in a database and look at the genes we want to target, pick out guide RNAs, and then they just order them from a company, complex them with Cas9 protein that we also order, and then we inject them into single-cell zebrafish embryos. Then we do laser injury and look to see whether they clot or not. In this case, we're looking at the involvement of platelets or thrombocytes. And so one gene we looked at, PLEC, normally when we do laser injury, this is a measure of how long it takes for the first thrombocyte to attach to the site of injury. And you can see in wild type or non-injected fish embryos, it happens pretty quickly within just a few seconds. But when we knock down this PLEC gene, so we reduce the amount of PLEC, you can see here that most of them don't attach in a 2-minute period. Also, we looked at the number of thrombocytes that attached in over a 2-minute period. And you can see that in the wild type, it averages about 5, and it ranges from anywhere from 1 to 10 thrombocytes, whereas PLEC, most of the time, none adhere at all. And then we looked at the number of thrombocytes circulating, and you can see it's equivalent. So it didn't do this by affecting the count of thrombocytes. It has an effect by affecting how well thrombocytes function, how well they aggregate. So this is one way we could quickly run through a number of genes and find how well they associate. And what was surprising to us is all the genes that we picked that I showed you, all of them were positive in these studies that we did. We looked at, this is looking at arterial clotting. We also looked at venous clotting, and they all had some positivity. And we're now studying those genes. And those genes are genes that we've never heard of before that have some relationship to, we've never heard of them as having any kind of relationship to coagulation. And now we found something. So that'll be very interesting to study further. So in summary, I've shown you that genome editing enables precise genome modification. It produces knockouts that we can use to model inherited bleeding disorders. It can repair disease mutations. It replaces the defective genes at a precise location. What I didn't talk at all about today is the risk of off-target effects. So it's a precise location, and these are chosen to hit that location. But what we haven't been as successful in is being able to prevent their promiscuity. So they will still hit other sites of the genome. And we can quantify those effects, but we don't know as well how to prevent that. And this can cause new mutations in other genes that can have side effects for gene therapy that could be problematic. So this is something that's always on the mind of people doing gene therapy. And I've talked to you a little bit about what we do in zebrafish. I've shown conservation of function, bleeding disorders. We've shown that they develop bleeding disorders similar to humans, but they have unexpected embryonic survival and adult lethality. And as I mentioned, perhaps this is something species specific to zebrafish, and that if we understand how that works in zebrafish, perhaps we can apply that to humans to treat patients with bleeding disorders. So thank you for visiting. I like to show this. I just discovered this in Michigan just 2 years ago. I've been in Michigan for 23 years and didn't know that there was a Jordan River and a national fish hatchery named after me, of course. And this is my lab group. We have positions available for postdocs and graduate students. Our division of pediatric hematology also has a clinical coagulation fellowship, if you know anyone interested. And I'll just leave you here with this video of Crispr fish. You can see they have no pigment at all, so they're entirely transparent. So thank you for your attention, and I'd be happy to take any questions.
That was fantastic. Let me go ahead real quick.
I
stop
my share. I tried to.
Yeah, I think we're all set. Okay, just as a quick reminder, nominations for Awards of Excellence are open through May 5th. If you go to hemophilia.org/BDC, you can head to the program tab and click on Awards of Excellence. From that page, click on the button for the nomination form. Self-nominations are not allowed at this time. You can always ask someone to nominate you. But if you know anybody that you would like to nominate for the Awards of Excellence, please feel free to go ahead and do so before May 5th. Okay, so I have a couple questions for you. Right. My first one would be, part of this research was supported by the Judith Graham Pool Postdoctoral Fellowship Award. How did this award impact your research?
Yeah, so, well, it actually, technically, it wasn't supported by Judith Graham Pool. It was supported by the NHF Clinical Fellowship. But it has supported, I applied for both at the same time and was awarded both, but could only choose one and chose the Clinical Fellowship. But the funding from NHF has had a big impact on me. And I can tell you that Judith Graham Pool specifically has had a large impact on other people. And what it is I actually didn't include slides I thought about including, funding for research, especially basic research, which I told you is so important to come up with things that maybe we never would have even thought of or tried to target, like CRISPR. That Judith Graham Pool funds basic science to do that, and it comes at a particularly vulnerable time for people. Usually, people who do their training have funding up through either graduate school or medical school. But when it comes time for that postdoctoral training after that, it can be hard to find funding and to continue on that research. And so having that funding was key for me to be able to stay in the lab and not having to do, too much clinical work, being able to limit my clinical work to a defined amount so that I can continue to do research. And that's been key for many people.
Great, thank you. You have worked with zebrafish for many years. Did anything from this most recent study surprise you?
Yeah, well, the main thing is what I talked about is the fact that the fish survive what is lethal in mice and presumably lethal in humans. Humans, we talk about factor 10 deficiency, factor 5 deficiency, they're incredibly rare. And I think the patients who have it, I don't know that there's any documented patient who has no factor 5, no factor 10, no prothrombin. Antithrombin's another one, no antithrombin. Those are all lethal. And in fish, they can survive into adulthood. And that was shocking to us. We have another loss of antithrombin leads to a syndrome called DIC, disseminated intravascular coagulation, which is fatal in humans if not treated. And they could tolerate that for a couple of months. And so, as I said, if we can figure out why fish can tolerate that, what's the mechanism, then maybe we can adapt that mechanism to humans.
Fantastic. Let's see. I guess you basically answered the next question that I have for you, but I'll present it anyway. Is there more you're hoping to discover in the future from working with zebrafish?
Yeah, absolutely. Right. So the things we want to do is get at modifier genes. I didn't talk about the modifier gene studies that we've done where we have had some success at finding genes that modify deficiencies of coagulation factors. These are done through genetic screens, that chemical mutagenesis I mentioned. We did a smaller one, and now we're actually in the middle of a huge one that's much more intensive. And then the other thing is to find new therapies. So I talked about those small molecule screens where we put a different compound in a well with a different individual zebrafish embryo to look for treatments. Love to find and these are oral medications. We're using FDA approved medications it's called drug repurposing, to try to find ones that might treat a bleeding disorder that are doing something completely different. And so, for example, doing that screening, we found that estrogen can cause thrombosis in fish. When we were trying to find procoagulants, and now we're doing a screen to find things that prevent that thrombosis using, again, FDA approved drugs to see if we can find a treatment for that. And we found a number of drugs that will reduce that thrombosis.
Wonderful. I have it's a comment, but I'm going to present it anyways. Nice overview, simplified it well, and thank you for showing lens clot. Let's see.
Now, and I see that was from Prasad.
Yeah.
Thank you, Prasad. I always like to show a picture of my host.
There you go.
And I show that.
So what were some challenges that presented themselves along the way in this study?
What were some of the challenges?
Yes.
Yeah, the biggest challenge was getting the genome editing to work in fish. And not so much to get it to work in fish, but getting it to work for us. Back in 2008, when I was it was around 2009, I was starting up my own lab. I really needed to have these models of bleeding disorders to do the modifier screens and drug treatment screens I wanted to do. And zinc finger nucleases had just been shown to work in fish, and they were very hard to, as I mentioned, very hard to make. We were trying to make some of our own. They all failed. We made about 20 of them. They all failed. And we got one to work out of the 20. And that was only one that was designed by a scientist named Keith Jung at Massachusetts General Hospital. And Keith was instrumental in getting genome editing, all forms of them. He helped us with zinc fingers and talons and Crispr and getting it out to what I call the masses of us. And without, someone like him who was working on this basic science aspects of it, without him, we wouldn't have been successful. And so that was the biggest hurdle. Once talons came out and he showed that they could work well in fish, and we were able to recapitulate that in our lab, it just opened the doors. It was a floodgates. And then when Crispr, it was even more. And since then, we've been able to do almost anything that we want.
Great, thank you. What is the biggest takeaway you want the audience to walk away with from your discussion today?
Well, I guess the general takeaway is the importance of basic science, and how, studies in something, seemingly as strange or, weird as a fish or studying immunity in a bacteria, right? You usually think about studying immunity to fight bacteria, but not the bacteria's immunity. That studies like that can be used to treat disease, and that we need to make sure, tell our government, tell your representatives that we need more funding for the National Institutes of Health and in the National Science Foundation to study these processes. And I see there's a question. Do you see that?
I do, yeah. Do you have an answer for it?
Yeah, so I'll read it. Yeah, so this is a question from Tanzania, Africa. Thank you so much for attending. And the question is, gene therapy is among the high, is a high cost of treatment. How is this going to help patients, especially those living in places like Africa where you can't get treatments? This is a great question. And my understanding is that I've heard that some of the companies developing gene therapy are committing resources to do gene therapy in places like Africa. Since some gene therapies, hemophilia A doesn't seem to be as durable, but hemophilia B gene therapy seems to be long lasting. The idea that this could be a one and done treatment, right? So simply you give the infusion to them and then it's done without needing to consistently get factor or, other kinds of therapies, repeatedly over years and years, right? That would be revolutionary and groundbreaking. The big challenge, of course, the price tag in the Western world is $3 million per treatment. So the challenge will be getting the funding. So it's still got the high cost, but I think in the long run, it will be a cheaper therapy. That would be great. Regarding the second question is regarding biobank. No single biobank in Africa. Is there any considerations or challenge to not being established in Africa? That's a very important question, and it impacts the Western world, and people are recognizing that more and more. While there may not be biobanks, the kinds of studies that these are doing are, originally largely focused on populations that ended up being Caucasians, and so more and more these large genome-wide association studies are called. Are focusing on other ethnicities around the world, right? So, and in the U.S. or in Europe, looking at people of African descent or people of Asian descent. So it's slow going, but it's moving towards that. In terms of actually establishing a biobank, it's hard for me to comment on that. It's a challenge. It has to do with money and systems, and right. It's not just money. It's the systems in place to collect samples and to bank them and all that, which takes money. So it's hard for me to comment on how close we are to getting there. I see there's another question, but that's just for you, Alson.
Yeah. I'll address that later, but you can watch this webinar back on YouTube or on our website for anybody else that wants to go ahead and watch it back. There's a lot to learn here. And I think that's it for us today, which was fantastic. Thank you very much for sharing your presentation and offering us your time this afternoon. We always appreciate your time and your expertise.
Thank you. It was my pleasure. It was a lot of fun.
Yeah, and thank you to everyone joining in today. Please note that this recorded webinar will be available on Friday, February 24th at hemophilia.org under the events tab with all of our archived webinars. Like I mentioned, you can also watch this back on our YouTube channel. Also available in the events tab is our upcoming schedule for our weekly Wednesday webinar series. Thank you for joining us this afternoon. Have a wonderful week, and we will see you next Wednesday. Bye, everyone.
Bye.